Hello students, this is the first video of molecular basis of inheritance and topic covered in this video is the DNA, in which we will study structure of DNA, structure of nucleotide, structure of nucleoside, pentose sugar and nitrogenous base. So let's start. DNA, which is called deoxyribonucleic acid, is a long polymer of deoxyribonucleotide. It means it is made when a long chain of nucleotides are linked together. Now, what is a nucleotide? A nucleotide is formed by three components: a nitrogenous base, a pentose sugar, and a phosphate group. This pentose sugar is ribose sugar in case of RNA and deoxyribose sugar in case of DNA. Given here is the diagram of a nucleotide. So as you can see here, there is pentose sugar which is made by 5 carbon. Carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. On carbon 1 is linked nitrogenous base. Now this nitrogenous base is attached at carbon number 1 by N-glycosidic bond. On carbon 5th is present a phosphate group. So phosphate groups attached at carbon 5th and this is linked by a phosphoester bond. As you can see here, there is a pentose sugar, there is a nitrogenous base linked at carbon 1 and there is a phosphate group at carbon 5. So this becomes a nucleotide. So a DNA is a long chain polymer of this nucleotide. Now let us study structure of dinucleotide. Now what do you mean by dinucleotide? Dinucleotide means when a two nucleotide joined together, they form dinucleotide. Given below is a one nucleotide and here is the second nucleotide. Now these two nucleotides join by phosphodiester bond. They join by phosphodiester bond. Now how is this phosphodiester bond formed between two nucleotides? You know that at 5th position phosphate group is attached, which we have discussed earlier, and at 3rd end OH group is attached. Now between this 5th phosphorus and 3rd OH, there is removal of water molecule. As you can see here, this is the removal of water molecule and the bond which we get is phosphodiester bond. Now as it is between 3 OH group of 1 sugar and 5th phosphorus of 1 sugar, it is known as 3' to 5' phosphodiester bond. So two nucleotides are linked by phosphodiester bond. Now let us study what is nucleoside. A nucleoside is formed when nitrogenous base is linked only with pentose sugar and there is no phosphate group. So we can say that nucleotide minus phosphate is equal to nucleoside. So given below is the structure of nucleoside. As you can see here, there is pentose sugar which is linked with a nitrogenous base. Now let us see the example of nucleoside. Adenosine, guanosine, cytidine, uridine are nucleoside of RNA, whereas deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine are nucleoside in DNA. Now let us discuss about nitrogenous bases, which is attached at carbon 1 of a nucleotide. There are two types of nitrogenous bases, purines and pyrimidines. Purines include adenine and guanine, whereas pyrimidine includes thymine, cytosine, and uracil. You have to remember that in RNA, uracil is present in place of thymine. So in place of thymine, uracil is present in RNA. Now let us discuss about pentose sugar. We already know that in RNA, ribose sugar is present, whereas in DNA, deoxyribose sugar is present. Now, what is the difference between ribose sugar and deoxyribose sugar? The difference is that at carbon 2, the ribose sugar contain OH group, whereas at carbon 2, the deoxyribose sugar contain only H group. That means oxygen is removed. That is why it is known as deoxyribose sugar. So if you have gained something from this video, please subscribe now to my channel Ramji Chopra, comment, like and share.